It's an old rule. But I love how you're trying to get out of presenting. Are you
Next
gonna
time.
rope in more people as we go along?
I have another student that will join, so maybe.
Maybe I'm gonna shift the entire schedule and
Okay.
Yeah, we also need to, the week we have the workshop in Bordeaux, we also have to cancel that one
yeah,
because
so that one is I'm gonna share the file again in the chat.
we're all there,
But
so we have to cancel that one.
so next week we're gonna have Hervé. The week after is gonna be Bordeaux. And then I'm gonna
Right,
have the
we'll
London.
fix that offline because it's time to get started. Good to see yours. Lovely to see so many familiar faces from before. Welcome back. Today we gonna Neuroccino with Fernando, who is gonna talk about, thanks for sharing the link, enhancing task fMRI individual difference research with natural stimuli. Take it away when you're ready.
Thank you very much, Steve. I shared the link again, just in
Yeah,
case for somebody that just arrived.
as if
And I'm gonna
anyone's
be
ever late to this meeting.
I'm gonna be
Eduardo
sharing
is not even
my
responding.
screen. Mm, yeah. Can you see my screen?
Yeah, we can. If you zoom in a little bit, we can see it full screen. Fantastic.
More?
Thank you.
Good. All right. So today it is not a published paper, it's a preprint. So take it with a grain of salt. But I found what they bring already very interesting. I think Steph shared this already a few months ago, this paper on LinkedIn, and I wanted to explore it a bit further because it's about quite a hot topic, which is the reliability. This test-retest reliability, the study of inter-individual differences, um, which has been shown in fMRI to not be that as strong as we think. So imagine if we would like to be studying at an individual level, maybe what if the patterns of brain activity that we see are reflecting better or worse performance. It's a bit difficult nowadays to explore this in fMRI. So what this paper is talking about already in the introduction makes a very nice introduction in the first paragraph about summarizing that fMRI is pretty much a growing field, and it's been more and more present in research lately, and more particularly task-based fMRI. But as I was saying before, there's been this problem in reliability. It seems to be less reliable and stable than initially thought. And this may be partly because fMRI data is slightly noisy, but also because there's variability already within individuals. And maybe the tools that we have nowadays are still evolving, so we're still getting there at the level of studying individuals. What we already have, and is quite powerful, is studying at a group level. So we can average these individual effects. And even though individuals may be varying within sessions, all the participants would stay within a specific range, within a specific average. So I'm not trying to say that fMRI studies are not leading to any conclusions. Just saying that at an individual level, there's still work to be done. So this paper is still bringing this up in the introduction in a way that maybe univariate approaches versus multivariate approaches may bring an advantage. So univariate approaches is where, for example, we're comparing one region with another one at a time, and then we find We try to find correlations there, or make conclusions, draw conclusions out of that. But on the other hand, with a multivariate approach, we're trying to find patterns within the data that can give us a bit more information. So, for example, by using a PCA, we can extract different ways in which the data can inform us in a more powerful sense. And an example of a multivariate approach is neural signatures, which is what this paper is talking about. They introduce that neural signatures have already been used for research. The more robust example that has been recently used is for pain, which is showing a very strong reliability. So the neurological pain signature. But for other tasks, it still appears to be doing quite a good job, not as good as for pain. It's being used, the neural signatures for studying task-based fMRI. And what this paper wants to dive into is testing whether there's a working memory neural signature that can be derived from the data set of the ABCD study, so adolescent brain cognitive development, which is a very large data set. So let's dive into what the paper is doing about this. What is this neural signature all about? So if we go all the way into figure 1, we can see that this is a visual of the neural signature. It's basically which regions are being active, differently active at the 2 back versus 0 back. So this is basically for the n back task. Yeah, I don't think I should dive into the n back task, but they basically split the data in 2, and then they train an elastic net based neural signature. And basically what they see in this figure that it's kind of giving similar results with differently split data. I mean, what we also see is that these regions that seem to be more active in a higher working memory situation compared to a lower working memory demand, we can see the frontoparietal, the salience networks, the thalamus, and the caudate all lighting up a bit more, which corresponds with what literature knows about this. So what they're basically verifying here is that the model seems to kind of work in the sense of here's how they built the model, and different ways of verifying it. And then for figure 2, yeah, of course, this classifier, this neural signature classifier, is only useful if it's consistent. So they're testing it with a split half reliability measure, and they're showing that it's kind of stable across the wave. So the ABCD study is recording brain activity in participants at age 10, and then 2 years later. And what they show is that for each of the waves, the first wave, this approach of the neural signatures seems to perform better, at least at the level of reliability, than when testing for individual regions. So that would be like an univariate approach versus the multivariate approach. The neural signatures are in the blue dot, and the individual regions are the distribution shown in yellow, because some regions will be more reliably explaining, more reliably activated in working memory situations versus other regions. And we see a similar pattern for wave 2, so 2 years later. And furthermore, at the level of, in panel C, the difference score has much higher ICC than any individual region, especially for the comparison of 2-back versus 0-back. So yeah, we have already a comparison with other univariate approaches. Neural signature seems to be good in this in terms of reliability. What the authors did further in figure 3 is still comparing. Well, it's comparing the neural signature to 3 different measures of task performance. So basically the task that the participants were testing, the n-back task. Then they're also testing this neural signature with cognition and psychopathology. So they see they see something similar as in the previous figure. Tested in comparison to individual regions, the neural signature has a positive correlation with the n-back accuracy. Then there's also positive correlations with this is the NIH toolbox for cognitive measures. So it's testing the total composite, which is composed of the fluid intelligence and crystallized intelligence. We also showed the list, working memory. And what they see is that the signature correlates positively with global cognition. So there's like a beta of 0.31 and also positively with working memory. What we see on the other hand in panel C is that it correlates negatively with symptoms related to psychopathology. What does this mean? So when participants have a stronger or a pattern of brain activity that's more similar to this brain signature that the researchers defined, this is correlated with lower is correlated negatively with symptoms to psychopathology. So having these brain signatures of performance to working memory seems to be able to, could be used as a predictor to better cognitive measures or to lower psychopathological traits. And off to figure 4. Because here the authors zoom in on where in the brain these associations come from. So trying to verify if this somehow relates to what literature has been referring to up till now. In a sense, do the regions most important for the neural signature align with the regions that independently correlate to cognition and symptoms? So is this biologically grounded in the task network or not? And what they find is it's a similar to figure 1. They define the main effects of the task with the warmer colors. It's a stronger activation for the 2-back task versus the 0-back. And in the second, in panel B, is the regional associations with attention problems. So in warmer, it's a stronger activation versus colder colors. Some more negative association. So it's higher activation is linked to fewer symptoms, basically. And in panel C, they're showing the main effect versus the attention problems. So it's so it's a yeah. So each dot is a region, and at the x-axis, they're showing association with attention problems. Y-axis is the strength of the main task, and it shows a clear negative slope, which is the regions most activated by the two back tasks are the same ones that are most negatively correlated to the attention problems. So it's all going in this direction. Gonna skip for the interest of time because there's two figures left that they're discussing here. And here in figure 5 they compare the neural signature to machine learning approaches. What they find, well, they're basically comparing the effect sizes in panel A. They plot the standardized regression estimates, and then the for the neural signature and the elastic nets prediction models that are trained separately to predict each outcome. So for cognition and task performance, the machine learning models perform about as well, or sometimes slightly better. So that's mostly what this figure is about. So it's seems like, well, it is machine learning models perform slightly better, although quite comparably well to neural signatures. So that is mainly seen in panel B, I would say. So the unique variance explained. They run models including both predictors together, the neural signature plus the machine learning model as predictors of the same outcome. And yeah, they both predictors contribute to unique non-redundant variance. So it seems like the machine learning models capture complementary information. And going into the last figure 6, what they show here is mostly that to train the models, even though the machine learning models may perform slightly better, the neural signatures that we have been seeing now require less training data to perform, to have a good enough performance. And this was what is mainly shown in this figure. So they're showing that the area under the curve at around a sample size of 320 becomes already starts to stabilize. And we see that in machine learning models that takes a bit longer, because machine learning models is here in the figure. It's in red compared to the neural signature in blue. This curve is stabilizing more around the 1280 mark compared to the 320 in neural signature. So there's a reduced amount of data that's needed to train this model, which could be useful for not only smaller studies, but yeah, it already becomes reliable after a few hundred participants, which is highly practical. So overall, they bring forward this case that these neural signatures that somehow capture these patterns in the data can be useful and can be thought as a latent factor, a genetic model that separates conditions. So they can be studied for not only for attention, but as said at the beginning, maybe studying patterns of brain activity for pain or more emotional tasks or different aspects of cognition. But it is an interesting solution to bring forward to this low reliability that the field is experiencing. So even with given the data, if there's no way to improve the data, which also improves the reliability, we can still do things with the data where we can extract more information. And I think this is the nicer part of the conclusion. I'm going to finish with this one. So they're making the point here that the multivariate approach is more reliable than the univariate approach. So comparing any individual region with itself is not as strongly reliable as with this neural signature. And that's pretty much it. It's yeah. There's a last point, which is it's probably not only that research can use this neural signature, but but it still remains an open door that it could be combined with other approaches that are multivariate as well, because there's been also other studies that model with multivariate approach how individuals vary over a session. They have just differences in performance. They perform better at some point, and then 1 minute later, they're not performing as well. And this variability can be modeled, and it also increases the reliability of being able to study at an individual level. So I wonder to what extent can it be combined? And who knows, maybe make this association that we have seen in one of the figures with diffusion-weighted imaging data. So yeah, that's it. Hope there's enough time for a question round.
Yes, you squeezed the time a little bit, but thanks so much for the presentation. Before I give the floor to the others for questions, just a point of clarification because I missed it and so did others in our group. What they mean by neural signatures is just the multivariate pattern of brain activation over individual regions activating, right? Is that correct?
Yes.
Okay.
So they trained this elastic net, which is it's basically capturing which are the regions that are most active during working memory, and then this becomes this neural signature, and the more
As
similar
a group rather than individual regions, I'm
yeah.
guessing. Yeah. Okay, cool. Thank you. Lilith has the first question for you.
Thank you, Fernando. It's pretty related to the previous question. So there was a figure where you're comparing machine learning with neural signatures. I didn't quite understand what is the difference if both are machine learning approaches. So what do they mean because neural signatures also you now mentioned that it's based on elastic net, that is a classifier that created two groups of patterns, and then they associated it with different cognitive performances and so on, as I understand, right? So,
Yeah,
and learning what? What do you mean? Like without grouping and directly just training on raw data and trying to predict individual, like skipping the grouping procedure or what?
mhm. Yeah, I haven't actually dived into this into too much detail, but I also wondered at some point, same question as you. I feel like maybe they're comparing with research that has already been done, or another machine learning approach that seems to be a bit more focused on the how to say classification of each of the cognitive measures. And I have the feeling that this machine learning approaches may perform better because they may be trained particularly for a specific kind of purpose, whereas the neural signature is somehow not necessarily purpose for that specific classification for the working memory tasks. I don't know if I'm making much sense now. And
So like
for you,
basically the neural signatures is just a type of working memory, right? I mean, you just group people into two types of quality, I don't know, of working memory.
yeah. So based on this data set where the participants were doing this emotional M-back task, they used this data to create the neural signature for working memory in this case. Yeah.
And then they just, but why they decided that the working memory groups will be associated with the rest of everything, basically, right? They just tried it,
Yeah.
or they had a
So
hypothesis
once they
that
had this pattern, they said, okay, is
what
it useful? Can we do something with it? Can this pattern, yeah, can it be useful to predict all this kind of stuff? Can be used for in the sense of cognition, can be used for predicting accuracy at the two-back task.
I guess one of the things to consider here as well is that the NIH toolbox is quite a superficial cognitive assessment in the end. So things like the MBAC task are quite reliable in terms of the cognitive stability, whereas if you look at the other measures, it does get a bit more difficult to predict those. So I guess that's also why the MBAC task is quite a good one to choose here because it's quite a strong effect usually.
Yeah, but I just don't understand the theoretical connection. Why would that be chosen to associate with the rest of cognition and also clinical pictures? As I understand, there was also like depression at some point, right?
Oh, yeah, psychopathology.
So yeah, it felt like they just, okay, let's choose this one and then classify it and then associated with everything else. So maybe I missed it. Maybe there is some more like theory
Yeah.
behind why end-back task will be a predictor of everything.
Yeah.
So
I think in my head, a point that they're trying to make in this sense is that I'm not sure if the brain works with neural signatures, but at least when the patterns that activity are so when people show activity that is more similar to this pattern that they derive, for example, for attention, they seem to also be correlated with better performance in these other cognitive measures or negatively correlated with psychopathology. So somebody that's showing more stable patterns of neural activity will also have lower traits of psychopathology in general. Although the negative correlation is not very big, but they're trying to point in this direction. There may be something in this stability of patterns of brain activity and maybe studying the brain towards the future with the lens of patterns of activity.
yeah, thank you. Didn't see the time. Then Leah, you go.
Oops.
Yeah.
I was enjoying the conversation. Now, I had like a question about why this data set. Like, is this a specific reason why they chose the ABCD study? It's kind of neurodevelopment. It's really complex, study the brain at that stage where it can change. And maybe you also have more variability, I guess. So maybe this stage of the brain development is gaining more from this pattern approach. Is there a specific reason why they are testing these in these brains and not in the adult brain that can be more maybe stable? I don't know.
Hey, yeah, thanks for the question. Yeah, I think I haven't thought of why, but now it makes fully sense that it would be if there's a data set to use, ABCD would be a great challenge for their approach, because you would expect maybe the most variability if you're using a sample that's adults compared to children, teenagers that they're using here. If there's any group that will be changing more, it's gonna be the younger population, but they still nevertheless find this stability within the patterns across time. So I guess it's a good choice, good that they make this choice, and it's also a happy ending for them because it turns out to be supporting their approach. But yeah, I think it goes in that way. Like I don't know whose idea was it, but I think it's a great idea to use this one, and it's also a very big data set, a lot of information. So
I was just going to say, isn't part of it the sample size?
as well.
Yeah.
I mean, for training a machine learning approach model, I don't know if there are other, how to say, other big amounts of data for adults or something like that with bigger sample sizes, but ABCD is the big one.
I see, because also like people studies the ABCD study also for pathologies. Like here, I try to read the methods, but I couldn't see if they are healthy or. But also you see these training performances in one of your in the figures that you showed. So yes, I was a bit curious about the data set in general and how can you have some biases, some like pathologies there. I don't know that are not diagnosed yet.
Mm-hmm. I think in general it's healthy participants, but I don't know right now if because they probably compare this to cognitive measures, so they may have done certain tests, certain behavioral tests that will point in this direction to be able to quantify the fluid intelligence or the working memory.
But there was a group with like attention deficit or something.
Yeah, indeed.
I think what they were trying to derive there is more the areas that would be more present in attention deficit kind of, well, the regions, sorry, that may be more present in attention deficit context. But I don't think they derived that from any part of the sample. I tried to dive into that, but I couldn't find the answer.
Thank you.
Thank you.
Thank you so much for the discussion. That's all the time we have. Thanks for joining us today. We have a CNS talk this week on Wednesday, 4 p.m. Amsterdam time. Looking forward to seeing you all there. It's the same link. And have an exciting week in science. See you next Monday, the latest. Bye.